If you can't make the lecture, it'll be recorded, and I'll be sending it to you guys by YouTube. My YouTube page is public, so I think you can just get on it anyway, but I've gone ahead and sent you the link. Someone actually just had a question for me. Oh, they just said yes. Okay. All right. So let me just go through what we're going to cover today or this evening. Hopefully you guys have had dinner, and sorry about taking you away from your families. So today we're going to cover Nervous and Sensory 1. Last time we covered an introduction to physiology. We'll cover a little bit of comparative stuff with animals, but just general nervous system function. Tomorrow we'll cover some more comparative, looking at different nervous systems, different behaviors, and different types of animals. Then on Monday, which will be the next lecture, so since it was kind of a short week, we'll start covering muscle. And if you take a look at the syllabus, the end of next week, we have our first exam. I'm planning to send out, first of all, an update of the first muscle lecture. I haven't posted it yet, but I'll have it to you by the weekend. And then I will also be sending out a study guide to you guys as well over the weekend. So let me go ahead and erase this stuff. Oops, what did I just do? Oh, well, hopefully you guys saw what I was just doing. Okay. All right, so let's go to today's lecture, which is the nervous system. And I'm sure many of you guys most of you are probably juniors, seniors. You understand some aspects of the nervous system. So some of this will be somewhat introductory, but we will be going over some different aspects of it, which will be kind of refreshing for you guys, especially as you're going to be applying to other programs soon and be taking entrance tests, things like that. Okay, so today we're going to go over, the topic is really on neurons and action potentials. Neurons, if you remember, of course, is another name for nerve cells, and action potentials really refers to the response of nerve cells when they're stimulated or excited. Much like yesterday when we were talking about feedback loops, right? When we spoke about negative feedback loops and also positive feedback loops, how cells respond to a stimulus, and in some case shut down that stimulus, in some case reinforce it. In the case of the nervous system, it's more of a reinforcement, kind of a positive feedback. All right, so to start off, I want to go over a bit of anatomy. Nervous system is made up of two distinct branches. The central nervous system, which consists of the brain and the spinal cord. And I'm obviously primarily referring to vertebrates, chordates, and things like that. Lower animals, lower levels on the evolutionary scale don't necessarily have it all developed like this. But our focus is going to be really mostly on vertebrates with this semester. So we have the central nervous system, which consists of the brain and the spinal cord. We have the peripheral nervous system, which consists of nerves that are going to and from the brain and the spinal cord. We call those cranial nerves and spinal nerves. Now, we can subdivide it further. There are some nerves, part of the peripheral nervous system, that their main function is to stimulate contraction or movement of the skeletal muscles. We refer to this as the somatic branch. Moving your arms and legs, writing with a pencil or pen, right? The contraction of those muscles is dependent upon the somatic branch of the peripheral nervous system. And you can kind of see that with this diagram. The other branch is the autonomic branch, and this involves control of our visceral organs, our viscera, our internal organs, our heart, our lungs, our digestive tract, our urinary tract, reproductive tract, immune system, all those are under the control. And we often refer to this as the involuntary branch, meaning a lot of the actions of the autonomic nervous system occur without our conscious awareness of it or control over it. Whereas the somatic branch, much of that is under our voluntary or willful decision. We decide that we're going to move our muscles. But as far as our digestive system, that's going to function, that's going to secrete enzymes, that's going to move food, whether we think about it or not. So that's why that's under the control of the autonomic. All right. So we can also, if we take a look at the nervous system, it's one of, at least teaching anatomy, I know it's most people's least favorite of all the systems. And one of the reasons, I think, is because it's not this nice little concise specific organs. You know, with the digestive tract, you've got the esophagus, the stomach, and the intestines. With the cardiovascular system, you've got the blood vessels, the heart, and the blood. The nervous system is just kind of all over the place. But we can take this vast system and really break it down into kind of basic functions. One of the things about the nervous system is I break it down into three. First of all, we have part of the nervous system I like to refer to as the input portion. Think of the nervous system as a processing of information. Just like a computer, just like a lot of things, you often have information coming in. In case of the nervous system, we refer to this as the sensory or afferent. And what do I mean by that? Say, for example, if you see an object, right, that visual perception, that's going to send information to the brain. That's sensory. Say if it's 11 o'clock in the morning and you're in the middle of class and you're used to eating and your stomach's growling, right? Well, information is coming in from your digestive tract, and all this stuff is being inputted from the peripheral system into the central nervous system, where it gets analyzed and interpreted. Once the decision is made what to do, we have the output, where nerves, actually either known as motor or efferent, send signals to specific organs. Say, for example, if our sensory, and we kind of put these two together, say with our sensory organs, say getting back to vision, say if we saw a dog that was chasing after us, right? Well, the sense organs sends input to the brain, and the brain responds, sending signals to your somatic branch, which is going to stimulate your muscles. You're going to start moving. You're going to start running, right? Another set of nerves may go to, that would be somatic. Another set of nerves, autonomic, is going to send signals to your heart muscle. Possibly your heart might start racing. You might start sweating, right? So this is what we call the output or efferent or motor part. You can take any function of the nervous system, as complex as it can be, whether it's memory, whether it's executive functioning or anything, and you could always break it down into some aspect of inputting, processing, and outputting. All right. Now, like I mentioned, the nervous system is very complex. However, there are certain aspects of it that are quite simple, right? There's simple functions of the nervous system and more complex. One of the simplest nervous system functions is what we call a reflex. In this particular case, that's my dog, is the simplified withdrawal reflex. This is a reflex that occurs, and you can see it looks like this person put their hand on a nail or a tack or something like that. We've all experienced something similar to that, whether we touch thorns of a rose bush or you touch a hot stove, right? We have nerves, we have what we call sensory receptors that are found in specific regions, in this case, the hand, that when this person stepped on or put their hand on a tack, the nerves, this would be an afferent or sensory nerve that carries information into the central nervous system. This happens to be the spinal cord. Then we have an output, a response via the efferent or motor, which is going to cause this person to withdraw their arm, right? First, it's an unconscious thing. You step on something, you pull your foot, or you move your hand out of the way. So this is really simple. Sensory, motor. Now, occasionally we will have in between that what we call these interneurons. You can see this interneuron. This is typically between the sensory and motor, and that can really process the information. Some reflexes stay between the point of stimulus, the spinal cord, and the target. Sometimes those signals are conveyed to the brain, and in this particular case, the signal going to the brain would be the sensation of pain, right? Because what do you do when you step on something? It's like, or put your hand on something sharp, ow, right? Because it hurts. All right, so this is just the basic, what we call a reflex arc. Now, getting into some comparative stuff, this is a cockroach, and despite what we think, or it seems, in terms of nerve responses, cockroaches are really not much different than us. When we talk about those three parts of the nervous system, remember I mentioned we have a sensory, we have a motor, and we have an interneuron. Well, here is in red, we have a sensory neuron. This happens to be a very long motor neuron that's going down what we would refer to as really a primitive spinal cord. It's not a true spinal cord, we just call it a nerve cord. And then here we have the motor neuron in purple. So what type of reflex, what type of response are we looking at? This is what we call the startle response. And it's really, the response is not any different than what I just showed you. In the case of the cockroaches, the receptors or the ends of the sensory neurons are found in these little antennae or little hairs. And in this particular case, the startling has to do with sound. So cockroaches is still or moving around, and suddenly there's a sound that comes in. That sound activates these little sensory receptors or nerve endings that send a signal down the sensory nerve into the kind of this primitive central nervous system. And then it all ends up with this leg muscle, and the cockroach is going to start scurrying away, right? Different type of situation, right? We're looking at startling, but it's no different really than the withdrawal reflex. And you can really look at the temporal relationship between all three of these different responses. For example, if we take a look on the y-axis, on the x-axis rather, we have a time interval. And here we have the different aspects of this response. Here we've got, in this case, it can be an air puff or a sound. And you can see this goes up first. This activates the sensory neuron. The inner neuron doesn't start right away, right? Because it's waiting for the signal from the sensory neuron to activate it. And then the motor neuron is a little bit later. Kind of makes sense. It's further along the path. And this ultimately later on, once that motor neuron activates a muscle, so this is kind of like the somatic branch, it's going to stimulate muscle contraction of that leg muscle. So here I'm just trying to draw a parallel in terms of, even though we're talking about initially kind of like the mammalian nervous system, the basic fundamental responses are really similar in all different types of animal groups. All right. So the nervous system, just like any other system, it consists of specialized cells that are designed to carry out a function. Muscle cells, of course, they're specialized to contract, right? Intestinal cells are important for digestion and absorption. Here we're looking at cells of the nervous system that are specialized in this communication. And that's really what the nervous system is, right? It's a communication system that's picking up information from one part of the body, transporting that message to the spinal cord and or the brain, and then a decision is made what to do about it. right? The cells that are responsible for that, or really the main cell type responsible, is the neuron. The neurons are the structural, functional units of the nervous system that are responsible for all that sensory, interneuron, motor responses. So incoming, processing, and outgoing. Now you might think, well, okay, that pretty much takes care of the nervous system. Well, we do have other cells that actually make up a larger percentage of the nervous system, and those are the glial cells. We don't often hear about these much. Some books don't cover them thoroughly, but they're very important. Glial cells are supportive cells. They help to maintain homeostasis, help to nourish neurons, they help to protect them, they help to kind of move debris that's not supposed to be there. So they have a very important supportive role, and it's been determined that sometimes defects or malfunctions of glial cells can lead to a variety of different diseases, things like Alzheimer's and possibly even Parkinson's. All right, so let's talk about neurons first of all. So they're the structural and functional unit. They're non-mitotic. They don't divide, right? So the neurons that you have as a child, those neurons, you don't get any new neurons. Basically, they don't divide. However, what happens with age is you can lose neurons, right, as we get older. What do neurons do? They receive and transmit information. How do they do this? By responding to a stimulus. So when you put your hand on a nail, or when that cockroach felt that puff of air or a sound, it responds to some sort of stimulus, whatever the stimulus is, right? Now, the response to the stimulus is an electrical response. So think of an electrical wire. We're going to take a look at this whole response, but it's an electrical wire that travels down the length of the neuron. And then what happens, that neuron has to communicate with other cells, whether it's other neurons or other target organs. And it does this by releasing what we call chemical messengers. So I know a lot of this is old hat for you guys. Some of it is new, but it's good to kind of refresh our memories like I do every year. All right, the anatomy of a neuron. So a neuron, as you can see, doesn't look like any other cell in the body. It's very different. We have really 4 main regions to it. The first region, which is probably the most similar to a typical cell, is this area. This is known as the cell body. And this is where a lot of integrating of information, integrating and processing of incoming information to that neuron. Now, where does that incoming information come from? It comes from these branches that are known as dendrites. So I like to think, when we were talking about the nervous system, we spoke about input, processing, and output, right? Well, we can zoom in to the cell and take a look at nerve cells have that same aspect about them, where there's an incoming of information through these branches called dendrites. The more branches there are, the more information that can come in. It's processed in the cell body, and then the response is via an electrical signal that travels down this structure, which is known as an axon. The electrical signal travels down this axon and takes it all the way to the very end, where we have what's known as this presynaptic or synaptic, more commonly called, terminal. This is known as the synapse, and this is where these chemical messengers are found that will communicate with the next cell. So we have electrical information going down the neuron, and that's conveyed into another cell via chemical messenger. Now, not all nerve cells, not all neurons are the same. They have different shapes. We're not going to get into all the different aspects of them, but they all have some aspect of this feature of dendrites, cell bodies, and axons and terminals right here. One aspect of neurons that are different is some neurons have this layer right here, or this interesting wrapping of a specialized cell called a Schwann cell. A Schwann cell is a glial cell found in the peripheral nervous system that forms what's known as the myelin sheath. And I'm sure you guys have heard of the myelin sheath. Damage to it is a characteristic of multiple sclerosis. This myelin sheath provides an insulation to the neuron, and that insulation, just like an insulation of an electrical wire, allows for an electrical impulse to travel faster. Now, having said that, not all neurons have myelin. Not all of them need it. Autonomic neurons, for example, are neurons that don't have it, whereas somatic neurons do. So it just depends. All right. Now, I want to zoom in a little bit closer to this terminal. And again, it's known by a variety of names. It's known as the presynaptic terminal. I've seen articles that refer just to the synaptic terminal. Sometimes it's referred to as the presynaptic terminal because this whole area is what we call the synapse. And the synapse is the point of communication between a neuron and another cell. So going back for a second, this nerve cell sends down an electrical signal, and that somehow has to communicate with another cell, whether it's a muscle cell telling it to contract, whether it's another nerve cell, like a motor neuron, whether it's a heart cell. It has to communicate with it, and this is the synapse. Now, at this presynaptic terminal, it's characterized by a couple features. Number one, we have these very large accumulation of vesicles, these spheres that contain neurotransmitters. We're going to learn about neurotransmitters. These are the chemical messengers that are found in the nervous system. You've heard of some of them, I'm sure, things like acetylcholine, dopamine, norepinephrine, many of them. There's a lot of different ones that we'll be looking at. And we also tend to have a lot of mitochondria down at this end as well. So we have the presynaptic terminal. We have this is the postsynaptic. This is the next cell. This is the cell that's going to be receiving the neurotransmitter. And the response is going to be a electrical impulse. So what we're looking at right here is a zoomed in version, of right up here. So this would be like a presynaptic terminal. Those neurotransmitters are going to stimulate that dendrite of this cell. You notice there's a space between this axon and this postsynaptic, this dendrite of another cell. This is normal. This is a gap, and it's called the synaptic cleft. Think of the neurotransmitter kind of swimming across to the postsynaptic side. Okay, so I gave you a little bit of the anatomy of the neuron, again, the basic functional unit of the nervous system. Now let's take a look at the glial cell. And you're going to find the glial cells, it's a whole other ball of wax. This is a diagram which shows the variety of different glial cells. Some of these are found in the central nervous system. Actually, all of these except one are found in the central nervous system, and that would be the astrocytes, the microglia, the oligodendrocytes. Unfortunately, it doesn't show another one called the ependymal cells. We'll talk about that later. We'll talk about their function. So there's 4 glial cells in the central nervous system. And there's one, in this case, one glial cell of the peripheral nervous system, and that's the Schwann cell. All right. So as long as we started off kind of talking about Schwann cells, now let's take a look at it a little bit closer. Schwann cells are a glial cell. They're only found in the peripheral nervous system, and their function is they myelinate the axons. This kind of looks very similar to the dividers of a swimming pool that divide the lanes. This is this insulation that forms along the length of the axon. You notice that the Schwann cells, which are wrapping around, and this is actually a Schwann cell nucleus that's just wrapping. You notice that it's not wrapped around the entire length. There's these little uninsulated gaps called the nodes of Ranvier. We'll talk about those later and why they're so important. All right, so we've got the Schwann cells. Another very important function of the Schwann cells is they produce neurotrophins. Neurotrophins are very important. I would call them a growth factor, that they're very important in the repair of neurons in the peripheral nervous system. The peripheral nervous system contains these Schwann cells, and what happens is if a peripheral nervous system axon is damaged, the Schwann cells produce these growth factors that helps in the repair process or the healing. This doesn't occur really in the central nervous system. That's why typically central nervous system neurons, again, do not repair, or is very minimal. That's why if someone has brain damage or spinal cord damage, the repair is very difficult. Although I'm going to mention some interesting research that's being done on spinal cord repair that can possibly overcome that problem. But neurotrophins are produced by Schwann cells. Hold on one second. Okay, Rai, you want to say hi?
I do want to say hi.
All right. All right, you guys, we're going to take a pause right here. My 15-year-old son wants to just say hi. Oh, let me just.
What's up? Yo.
Yeah. All right. See you, dude. All right, let me go back to where were we? Okay. All right. So that's Schwann cells. They make myelin and they produce these growth factors that stimulate repair. Now, when we talk about myelinated axons, as I mentioned, this is just another view of it, showing how the purpose of the myelin is that it speeds up the propagation of an action potential. An action potential is that electrical impulse. So with the myelin, the electrical impulse gets here quicker. Without myelin, it gets there slower. And again, it just depends on the neuron. All right. Now, one of the things when we talk about different animals, an excellent model for studying the nervous system is the squid. And if you look at some great animal physiology books, virtually every system in the body has an excellent animal to study it. For example, the reproductive system, a good model to study it are cattle, because cattle have a 9-month gestation period. In the case of neurons, the nervous system, squids are a very good animal to watch to study neurons, because they have a giant axon that it's much larger than anything we have. And they're also unmyelinated, okay? This is a cross-section through a squid. These are normal-sized axons, and it's kind of hard to picture, but it looks like empty spaces, but these are all individual axons kind of coming out towards you. And then in the mix, we have this giant axon, and this plays a very important role in movement of the mantle muscle. If you've ever seen if any of you guys are divers or just seen on any National Geographic shows, when squid are startled, they contract those mantle muscles, and basically they get this kind of a jet propulsion movement, okay? So they have large unmyelinated axons, but we have very small myelinated ones. These small little areas, so this happens to be a nerve, but a nerve is made up of many axons. So each of these tiny little things are an axon. This is a giant axon. So in other words, we have many, many small myelinated axons. We don't have a giant axon. So why not? Why don't we have a giant axon like a squid? Well, first of all, it wouldn't be practical for us, right? Not to minimize some of the functions of a squid, but compared to us, a squid does not have to move and do a lot of the activities that we do, a lot of the manual manipulation, thought processes, all that. Squid has really limited functions compared to us, so it gets by with that. If we were to take that giant axon and try to put giant axons in us, we would have, it wouldn't be practical. We would have to be enormous. So through evolution, we've evolved, instead of having a giant unmyelinated axon, to having smaller myelinated ones. Okay, so large axons are not practical for us because it just wouldn't fit. For squid, they're fine. Now, I mentioned how myelin is really important for the speed of an action potential. Well, there's actually two factors that play a role in action potential velocity. Number one is there myelin or is there not? With myelin, increases the fast conduction. The other variable is diameter. Larger diameter axon also increases speed, right? So we don't have the large diameter, but we've got the small myelinated ones, which gives us a fast velocity. In this case, the squid with this one doesn't have the myelin, but it's very large. So it's able to kind of compensate for that. And I'm going to show you a really cool video at the end of this, showing the squid and how a lot of our understanding of the nervous system came from studying the squid. All right, moving on to the central nervous system as far as glial cells. You see we have four. The first one is the oligodendrocytes, and the oligodendrocytes are to the central nervous system what the Schwann cells are to the peripheral. They make myelin. We have astrocytes. Astrocytes got their name because when they were first seen under the microscope, they kind of had this star-like appearance. They are very multifunctional. They, much like Schwann cells, they do produce neurotrophic factors, especially during development, that helps with kind of growth of the central nervous system. Doesn't really help much with an injury, and I'll explain why. So they produce growth factors, they provide nutritional support for neurons, they recycle neurotransmitters. So neurotransmitters, once they have their effect, they're not just typically lost, they're broken down and reused. And finally, astrocytes make up what's known as the blood-brain barrier, which is a protective barrier between the blood vessels and the brain that helps to protect the brain against influx of most toxins, most fluctuating conditions from the rest of the body. This blood-brain barrier is helpful, but it can also be problematic, especially if someone has, say, brain cancer, and doctors need to get chemotherapeutic drugs into the brain. That blood-brain barrier sometimes will impair that ability. So there's a lot of interest now, research being done to find ways to kind of bypass that blood-brain barrier. Now, what about these neurotrophic factors and injury? So this is one of the key differences between the central nervous system and the peripheral. In response to an injury, the astrocytes begin to form scar tissue. And if any of you, I know everyone's had some injury sometime in their life where you get scar tissue. Scar tissue is typically a normal consequence of an injury, but eventually, especially in other parts of the body, it breaks away and the body heals sometimes, right? In the case of the brain and the spinal cord in the central nervous system, the scar tissue helps. What it does, it prevents the exposure of the injury to healing factors. So it's almost like a barrier, and that obviously creates a problem. Next cell are the microglia. Microglia are kind of modified cells of the immune system, kind of modified phagocytes. And just like phagocytes in other parts of the body, in the blood and in the lungs, for example, they remove stuff that's not supposed to be there. Debris, anything they're kind of like the housekeeper. Finally, we have the ependymal cells. And what it doesn't say here, but what's really important, is they help to produce cerebrospinal fluid. And they're also the source for neural stem cells. Now, what's interesting, we often hear about stem cells. That's a very kind of a hot area, different areas of science in terms of stem cells that can help to sort of replace injured ones for different types of illnesses. Pancreatic cells, for example. There are stem cells in the brain that have been used. Obviously hard to get to, but they are kind of self-regenerating. All right. Oops, okay. So now that we've kind of covered all the glial cells, can non-mitotic neurons regenerate? And I've already kind of answered that question. Yes, they can regenerate. They can be repaired if they're injured, really primarily in the peripheral nervous system as opposed to the central. What happens in the central nervous system, like I mentioned, the glial cells, the astrocytes, tend to cause scarring, which interferes with growth and repair. The repair of neurons in the peripheral nervous system requires neurotrophins from the Schwann cells. So let's take a look at repair of the peripheral nervous system. So neurotrophins, that's kind of like a general name. All it really means is kind of maintaining neurons. Well, there's a lot of different things that they can do. Neurotrophins are important during fetal development. That help in the growth and development of neurons while babies are still in utero and continue on once they're born. They're also important in helping to maintain function of adult neurons, kind of keep them going for a while, right? They're important in regeneration. So in addition to maintenance and development, they're also important in repair. And that's where regeneration comes in. There's two different types of growth factors that I want to mention that are used in the treatment and development of neurodegenerative conditions. Let me go to this slide for a second. Those two different growth factors I mentioned, we have NGF, and that stands for, very easily, nerve growth factor. And then we have BDNF, which stands for brain-derived neurotrophic factor. All right. Now there's others, but those are some real key ones that do play a role in repair. Even though it's called BDNF, it is also found in the peripheral nervous system. And in fact, there's a lot of interest now, and there's been some success in some labs from infusing or treating patients who have neurodegenerative conditions with some of these nerve growth factors. I mean, I don't want to overemphasize it. Obviously, there are a long ways to go, but there has been some success. This is a diagram showing an example of a neuron with the cell body, the dendrites, and the axon and its branches. This is actually in a petri dish. So this is a typical normal neuron. This is a normal neuron that's being treated with some neurotrophins. And look what happens. You notice the growth factor did not cause the cell to divide, but it just got more sprouts to it. And the more sprouts you have, the more information that can be processed. All right. So speaking of nerve regeneration, I just kind of wanted to go through the process of how neurotrophins work. Again, these are the neurotrophins produced by Schwann cells. Here's our neuron, our cell body, dendrites. Here's our myelinated axon. And this axon would be a motor neuron that's going to skeletal muscle. Well, say this neuron is damaged. The first response to any damage is the distal portion, so the part that's furthest away from the cell body. Yes.
All right, we can't see your slides.
Has that been the case the whole time, or?
No, just since you did the NGF nerve growth factors.
I know why. Well, thank you for letting me know. I know exactly why. Hold on. Yeah, I got to actually go to here, and then you see that?
Yeah.
Okay. Yeah, I'm still learning the tricks of this thing. Thanks for letting me know. Otherwise, I'd just be walking through this aimlessly. So the distal portion degenerates. And as it degenerates, you can see the connection with the muscle completely is broken down. It's basically, the body's doing this. The immune system and other cells, they're just removing this damaged cell, right? This damaged portion, I should say. We want to get rid of this damaged area. It's inflamed. We want to get rid of it. And then the Schwann cells kick in, and the Schwann cells begin to aggregate around the remaining portion of this axon and begin to assemble, secrete neurotrophins, and essentially guide it through kind of like what we call a regeneration tube, guide that axon back to the muscle. Now, how long that takes can vary. It just depends on the nature of the injury, but that's typically how many nerve injuries. And obviously, some severe nerve injuries don't repair, even in the peripheral nervous system, but many of them do. All right. Okay, so that's the anatomy. That is of the neurons, the glial cells. We know basically how the nervous system works. Remember, I mentioned there's an input of information, there's a processing, and there's an output. And I mentioned the way that this communication goes is primarily through an electrical signal, which then ends with a chemical signal. So let's take a look at exactly what happens, how the neurons respond. So first thing, before we talk about the response of a neuron, I want to mention what cells are like, what the inside of a cell and the outside of a cell are like when they're resting. And resting is it's a term that's used. It's not really correct. I mean, resting implies that the cell's just sitting around doing nothing, right? The cell's always doing something, even if it's like some minor kind of housekeeping stuff. It's meant to imply that this is when the cell is not being stimulated. So when a cell is in its resting state, if you were to take a voltmeter, because all cells have kind of a voltage to them, you would find that the inside of the cell has a negative charge, roughly around minus 65 millivolts. Some books will tell you minus 70, some books will say minus 60, but it's around minus 65, and this is known as the resting membrane potential. It's the voltage. Now, why is this? Well, of course, you've got the inside of the cell and you've got the outside of the cell. In the inside of the cell, first of all, you've got a lot of protein anions. Proteins that are negatively charged, that's what anions are. They tend to be on the inside. And are these green kind of negative charges, right? Now, we do have some positive charges. We have a lot of potassium on the inside, potassium ions. But everything else is on the outside. If we take a look, the outside has a buildup of sodium. And we do also have some chloride. My main focus today is really going to be on sodium and potassium, but at least I wanted to sort of kind of lay the groundwork to emphasize that the inside is negative relative to the outside in any resting cell. Now, you may know from other classes that ions do not normally cross membranes. They can't, well, first of all, they can't cross a membrane. The only way they can get past the membrane from one side to the other is through ion channels. And so we're going to take a look at some ion channels and how they work at rest and also when the nerve is stimulated. We're also going to take a look at the role of a very important active transport mechanism called the sodium-potassium pump. Now, as far as the numbers, I mentioned there's protein anions inside and there's sodium outside, potassium. Just to give you an idea of the numbers, kind of the relative abundance, potassium is 150 millimolar on the inside, only 5 outside. So it's really inside. Sodium, 145 outside, 15 inside. So it's really just the opposite. And right now we're not going to worry about chloride and calcium. I just kind of want to focus on those two, realizing that in a resting cell, we've got a lot of potassium inside, a lot of sodium outside, and we've got these ion channels. And this is just another diagram kind of showing roughly the amounts. Again, it's going to be a little bit different here, but inside, a lot of potassium, very little out. A lot of sodium outside, very little in. I'm not going to ask you the specific numbers, but I do want you to know which side of the membrane has more sodium outside, right? More potassium inside. Yeah, and you do need to know the voltage, though. Okay. So we have a lot of the sodium outside, I should say, and a lot of potassium inside. Now, you may have heard of what are called voltage-gated ion channels. And these are channels that open up when cells are stimulated to allow ions to move. In a resting cell, those voltage-gated channels are typically closed, are always closed. However, we do have what we call a leak channel, which is this right here, which allows potassium to leak out of the cell. So in a typical cell, cells are, even though we have sodium on the outside, potassium on the inside, the cells are much more permeable to potassium. And since potassium is higher inside, it's going to leak out. Okay. At the same time that the potassium is leaking out, and I'm going to show you kind of right here, there is some sodium that's also leaking in. Now, this is, again, in a resting cell, right? Even though sodium's high outside, potassium's high inside, we've got these these leak channels. And think of like a dripping faucet, right? We have this kind of a little drip of potassium going out, a drip with the dashed lines of sodium coming in. And the reason why sodium comes in is because there's more outside than in. It diffuses in, potassium diffuses out. But this kind of presents a problem, because if potassium continues to diffuse out and sodium continues to diffuse in, there goes our resting membrane potential, right? This minus 65, where we've got to have a lot of potassium inside, a lot of sodium outside. So fortunately, we have a mechanism, the sodium-potassium pump, or the sodium-potassium ATPase, which takes whatever potassium that came out, pumps it back in. Takes whatever sodium that came in, pumps it back out, and that's this right here. This is not only in neurons, most cells of the body have this pump. It's very important in restoring the resting membrane potential. Okay, so what we've just looked at is what happens when a cell is at rest. Now let's take a look at when a cell, or a neuron in this case, is stimulated. So I already mentioned about these leak channels, these leaking channels. Well, we're going to kind of put those aside for a second, and now we're going to focus on another set of channels called voltage-gated channels. These channels are closed when the cell is resting. They only open when it's stimulated. When a cell is stimulated, the first thing that happens is those sodium channels voltage-gated sodium channels open up and sodium floods in. This is known as depolarization. This happens first. This is followed, and it's a delay, by the opening of voltage-gated potassium channels, and potassium will diffuse out. This is called repolarization. It's an efflux of potassium, goes out. Now, what's interesting is there's kind of an imbalance, right? The amount of sodium that comes in is not balanced by the amount of potassium that goes out. In fact, more potassium leaves than sodium that comes in, so the cell becomes what we call hyperpolarized. And I'll show you what that looks on a graph. So this is the typical pattern of when a nerve is stimulated: depolarization, repolarization, hyperpolarization. Someone have a question? I know I heard some noise. No? Okay. All right, we're all good still? All right, I guess so. All right, so what I've just mentioned to you, this response, is what's known as an action potential. And this is an all or none. This is as a result of a stimulus that basically causes this depolarization. If we're going to look at this on a graph, this is a graph, and you may have seen this in some physiology labs before. This is the time, milliseconds, and this is the voltage. Remember I mentioned that the cell membrane, or the inside of a cell, is roughly about -65. In this case, it shows -70. It's about that. When the cell is stimulated, what happens? Sodium channels open, voltage-gated channels open. The sodium permeability increases, and as sodium comes in, remember sodium is a positively charged ion. So that's going to make the inside become positive. And if this response goes all the way to the max, the inside of the cell becomes about +30 voltage. And you can kind of look at the sequence of events here, right? Now, at the same time that the sodium channel is, first it was open and then it begins to become inactivated and close, potassium channels open. Potassium is also a positively charged ion. That leaves the cell, so now the inside is going to become more negative. And again, as I mentioned before, it goes past this -70, even below that. Even in this case, it looks like it's about -80. This is where it becomes hyperpolarized. And then eventually, of course, we return back to the baseline. So this is really the three parts of a typical action potential. Sodium channels open up first. They're just quicker. Sodium's first, potassium is a little delayed. They're a little sluggish. And then potassium channels don't close as quickly either. That's why we get this hyperpolarization. All right. This is just another diagram showing the physiology of it. Some of you guys might know Greg Harris. I'm sure many of you do. He's done in some of his research what's called patch clamping, where you can look at movement of ions across through channels. And this is showing, here's a graph showing this downward deflection, which is represented by sodium channels opening up. And you can see sodium's coming in. You notice later on, these are potassium channels that are opening up. And then it all comes back. So where it's flat, right, nothing's open. That channel's not open, but when you see this change, this is indicating an opening of sodium channel. Sodium comes in. This is opening of a potassium channel, which occurs later. Potassium's leaving. All right. Okay, so a little bit more about channels. Channels are really the key to all of this. And interestingly, clinically, there's a lot of different drugs and medicines that are used that either block or stimulate the opening of channels. And we'll talk about that shortly. Now, channels can be in one of a couple different states. It really depends upon the ion. most channels, for example, like potassium channels, voltage-gated potassium channels are either open or they're closed. Sodium channels are either open, closed, or inactivated. All right. If we take a look at a typical response, an action potential, here the channel is closed. Here it's open. This is, I'm sorry, this is, here it's open. This is B. Stays open. Then it becomes inactivated and then eventually closed. So it goes from closed to open to inactivated to closed. Potassium channels only are open or closed, right? In reference to the other channels, and this only happens, this doesn't have all of them, but just to kind of reiterate a little summary, potassium leak channels are always open, right? They're always going to be some leakage. Same thing is true with sodium leak channels. Doesn't show sodium leak channel here, unfortunately. The voltage-gated channels are only open during the action potential. When a cell is depolarized, the sodium channel opens up. There's the voltage-gated potassium channel. It's still closed, right? Here, the sodium channel is inactivated and then eventually closed. Here, the potassium channel is closed. Then during repolarization, it's open. Then during hyperpolarization, it's still open, but it's starting to close, and we're starting to get the movement of potassium back. But this is meant to illustrate kind of the different behavior of the channels. All right. So I want to kind of readdress something that I mentioned last time, and that was feedback loops, right? We spoke a lot about the theme of physiology is homeostasis, a very important central theme. And this homeostasis is governed by negative feedback, right? But I also mentioned there's positive feedback, which is a bit more of a sort of a short-term event. I mentioned things like, for example, childbirth, right? Oxytocin production, onset of labor, that continues until the baby's born. I mentioned blood clotting. Another positive feedback is our action potentials. We have sodium entering, we get depolarization, sodium enters the cell, which triggers more depolarization, which triggers more sodium channels opening. This is a positive feedback until the sodium channel becomes inactivated. And then we get potassium channels opening up. So just to kind of reiterate the application of feedback loops to this. All right, let me just see how we're doing here. Oh, wow, we are moving along. Hold on a second. Yeah, again, I want to thank you guys for showing up. It's such a weird time. All right, so that takes us through kind of a lot of the introduction to the nervous system in many cases. And next time we're going to, on tomorrow, normally it would be every other day, but tomorrow we're going to cover a lot about synaptic transmission, the chemical messages, and how we see this in different types of responses in sense of smell in many animals, how the olfactory response is neurologically based. Before I get to that, I do want to talk about an interesting aspect that really applies, especially since we live along the coast, San Diego. That has to do with certain toxins and how they affect the nervous system. There's a toxin called saxitoxin, which is produced by algae that occurs during red tide. And I'm not sure if some of you guys had a chance to see the red tide. I'm not even sure if it's still going on, but then at night you see the bioluminescence. It's really cool. But some of the algae can be very toxic. And with the typical food chain, the algae, and this is the one algae we're talking about here, this Alexandrium catenella, the food chain, the algae is eaten by shellfish. And of course, shellfish is eaten by mammals, including us. Well, saxitoxin happens to be a sodium channel blocker, right? So we know the importance of sodium channels and the influx of sodium in a nerve response. So when you block that channel, you can affect a lot of things. You can basically lose sensation, like a tingling or a numbness at the minimal, or sometimes around your mouth. In severe cases, depending on how much is consumed, someone can die from respiratory failure because of the effect of the nervous system, especially the sodium channels and muscle contraction, diaphragm muscles, things like that. Now, eating sushi, we all love sushi. I think I love sushi, but it's really important that where you go to get it, make sure it's processed properly. Some of my students in the past have mentioned that they've had experiences of having shellfish where they got this kind of numbness around the mouth. So it's an interesting application of it. All right. I'm going to primarily end this talk, go over a couple slides with showing you a little bit more of the squid axon, giant axon, I should say. So this is actually, this is the squid. Within the squid, we have, these are these mantle muscles that we'll be looking at shortly, and the squid axons actually stimulate contractions of those muscles for movement. This is a close-up view of the squid where we have this very large central giant axon, which is surrounded by many small ones. We're going to take a close-up look at this giant axon in a couple slides so you can see a really neat video to understand. And this is one thing that's cool about this is that it's much easier to study something if you can see it really well. It's not microscopic. You can see it to the naked eye. It's easier to study. And that's true with the giant axon. Okay? So this is a YouTube video of the squid axon. This is actually a video that was made probably back in the 60s. You'll be able to tell from the just the sound and the way it's described and the quality a bit, but it's very informative. And just to kind of prepare you, it's a video that shows kind of some of the early investigations of the squid axon and how it was the studies of the squid axon that gave us information on understanding that how nerves function, sodium coming in, potassium leaving. All right. So I'm going to, hopefully this will work. Let's see. Oops. Let me pause for a second. Let's see. Can you guys hear it okay?
Not really. Also, we can't see it.
Oh, that's a problem. Okay, hold on. Yeah, you can't, if you can't see it, that's no good. All right, let me go back. Let's see, hold on a second. And share and click on this and share. Okay, can you see that now?
No.
What do you see?
The slide of the squid.
Oh, I mean, you see the squid, you see where it says YouTube?
Yeah.
Okay, now if I click on this, does the video come up?
No, it just brings up the slide.
Oh, wow. Okay, hold on a second. Let me see if I can correct that. All right, hold on. Let me see. I've never had this happen before. Okay, hold on. Let's see. That's bizarre. Let's see here. Let's do this. Let's screen sharing. Share. How's that?
Yeah, I can see it.
Okay. The sound's not that great, but do the best you can, and you should be able to still understand some of it pretty well. So. This is really cool. All right, let me get rid of this. Let's see. Okay. Oops. Okay. Let's see. Okay, so hopefully you guys were able to get something out of that. Again, if there's during the lecture, feel free. I know a lot of you guys are, shy or just don't want to say anything. If for some reason you can't hear me or see anything, just let me know. I mean, granted, you may have done that on the chat, but I just didn't look. But definitely feel free to just shout out, because I don't want you guys to sort of, not be able to see everything. All right, so he was saying, like the understanding of sodium channels led to the development of local anesthetics. So like lidocaine and novocaine, they're also sodium channel blockers. So something interesting to know. All right. Just to kind of go back and look at kind of an interesting comparison and how we're able to, why studying the squid axon is very different to looking at other cells in mammals and things like that. In this case, where it's a mammalian muscle, which is also excitable, but it would probably be very similar in a mammalian nerve. The outside always has more sodium than on the inside. Same thing here. The inside always has more potassium than the outside. Same here. So there's a nice consistency, and that allows us to really do this nice comparative physiology. All right, just a little bit more about action potential since we spoke about this giant axon. Remember I mentioned the conduction speed now has two different variables. Number one, myelination. If a cell is myelinated, if an axon is myelinated, the action potential will be faster than if an axon is unmyelinated. And then diameter. A larger axon will be fast, will have a quicker action potential than a smaller one. The reason why an action potential is faster in myelinated axons has to do with what we call saltatory conduction, where ions can only enter into the axon at these nodes. And the idea that it kind of jumps from one node to the next, kind of a saltatory conduction. This is, saltatory conduction is always applied to myelinated axons. Just to kind of give you an idea, if you think of when you take a flat rock and you skim it across the lake, how it kind of, if you do it just right, it kind of jumps. That would be kind of like a myelinated, an action potential in a myelinated nerve. Oops. All right, last slide is really related to that. And just to illustrate that across the animal kingdom, whether we're looking at squids, whether we're looking at fish, which is a teleost, whether we're looking at shellfish, a frog, or a cat, there's always going to be a correlation between axon diameter and conduction velocity. You notice the slope. It's always, sometimes the slope is steeper, but there's always going to be this correlation between diameter, increased velocity. All right, so the stem cells, okay, so let me just go over a couple questions. Do stem cells make like to self-renew, then differentiate? Yeah, well, if Daniel is still here, they have used neural stem cells in research, and they've been able to induce them to become, instead of just ependymal cells, they can become different types of, they can become neurons, they can become oligodendrocytes. I don't know if neural stem cells can become other types of cells, like muscle cells and things like other stem cells can be. Slides cannot be seen. Hopefully we got that corrected. No, maybe exit out full screen of the PowerPoint. Yes, perfect, okay. Is the video supposed to have sound? Thank you, okay. So we'll get through some of the bugs, everybody, but hopefully you were able to hear me. I assume you're able to hear me. Were you eventually able to see the video of the squid? I assume, yes? No?
Yeah.
Okay, all right. Okay, so I will see you guys tomorrow. Tomorrow we're gonna meet at noon. Again, everyone has different things going on in their life. If you can make it, great. If not, I'm going to record it and then send you the link. Tomorrow's Thursday. So sometime over the weekend, I will be posting the muscle lecture as well as the study guide for the exam. So there's no other questions. I will hopefully see you guys sometime tomorrow. Have a great evening.